Hello everyone. This section deals with mapping proteins to function. This section also is for mostly for the bioinformatics and for this and for what I will say, who does cheminformatics as well as bioinformatics together, and it's heavily concentrated on the bioinformatics methods and the molecular biology methods and everything. So to start with, what we learned earlier, like what are proteins? They are like unbranched chain of amino acids, and there are 20 different amino acids, and there are 4 different levels of protein structure: the primary, secondary, tertiary, and quaternary structures. And protein amino acid sequences determines its three-dimensional structure of the protein. The proteins have some functions. So using those functions, our body functions, our body works. So why we are studying protein function? Because these proteins are responsible for several diseases in our system, and for which we need drugs to cure it. And also, apart from that, we need to understand how the protein works, which means the mechanism of action of proteins. So as you can see from the diagram, like protein can function as in signaling pathways, and it's in transport, in catalysis, and in movement, and in regulation, and in structure. So these are the various types of functions inside our system, and it is the sole, you can say, it is the sole element. These are the sole elements for which we work, our systems work, and why we have diseases and everything. So to predict the protein functions, many scientists use amino acid sequences, protein structures, genome sequences, any phylogenetic data, microarray expression data, the protein interaction networks, and biomedical literature. So microarray expression data, we'll be talking about this in the next section, considering the protein interaction network. So for example, proteins and proteins used to interact in sense some proteins bind to the other protein and cause some kinds of signals to work on, or they bind and they work as a kind of a complex. So that is how the protein interaction networks are, and how we can assign functions to the proteins. And also there's text mining approach, where if proteins are appearing in a lot of biomedical literature, that means two proteins might be get connected. So it's something like that. So these are the data sets which are used to do protein function. But the concept of protein function is highly context sensitive, and it's still not very well defined clearly, because they act as a kind of umbrella term, like they are kind of hierarchically related. For example, the cellular function, the molecular, and the physiological function. A protein can have a cellular function, kind of have a molecular function, or have both cellular and molecular, which shows some physiological effects. So that's why the Borg it all classified the protein as you can see, classified protein hierarchically in terms of phenotypic function, cellular function, and molecular function. So the cellular function is the metabolic pathway which is involved, or where it is present in cytoskeleton. The localization, the molecular function is where it is binding or having some catalytic activity or having some conformational changes, and the phenotype, what the phenotype it shows, or what affects the system. For example, whether it is having some rashes or some skin problems or something like that. This is how the proteins actually kind of function. So based on the above, the 1998 paper, later on they have uh, classified the terms, the phenotype, the molecular function, and cellular component in a kind of ontology based pattern. The ontology aims to develop the control vocabulary of gene and the gene products, and it also helps to annotate the gene and the gene products and assimilate and disseminate the annotation data. So, and also provide tools for easy access to all aspects of the data provided by that protein. And the ontology can say it's kind of representing the knowledge of a set of concepts within a domain. For example, if you are a computer scientist, your domain is mm, computer science, programming, database development, or web development. So all these terms, all the knowledges, all these concepts comes into a single domain of computer science. So this is how what's the kind of ontology is, and the gene ontology is kind of divided into a cellular component, molecular function, and biological process. A protein is associated is assigned all these ontological classes, and then they used to functionally annotate the protein. You can visit geneontology.org to get the gene ontology terms, and you can just type in the proteins sequences or the names, and you can easily find the find the functional terms associated with the protein. So actually the GO is divided into kind of cellular component, molecular function, and biological process. And so what does it do? And it helps actually it uh, this all these terms has a very wide coverage, and it has a standardized format, and it has a directed acyclic graph-like structure, which is kind of hierarchically related, and the categories are kind of disjoint. And here it's pretty easy to assign functions. For example, the molecular function, you can see it's nucleic acid binding, then and divided into enzymes. So for example, anything which is been here, so they have been classified as this term here, then as this term here, and then this term here. So if anything changes here, this annotation gets automatically changes. The molecular function is the elements of all the elemental activities of a gene product at the molecular level. For example, the binding or the catalysis. Uh, the biological process is the operation of sets of molecular events with a defined beginning and end, like pertinent to the function of integrated living cells. For example, the metabolism and everything. And the cellular component is a parts of a cell or its extracellular environment. The cellular component are probably the best defined since they correspond to the kind of the actual entities. And the biological process and molecular function is kind of difficult to distinguish, but still, it's all the molecular function forms a biological process. So David is a very popular tool to assign functional functional classification of the proteins. You can watch some videos here, and it is a list of microarray genes where you can paste it on David and try to see what David tool does. The next part of this lecture will be based on sequence and structure based methods. So basically, annotating a proteins, we assign protein names. We do give gene symbols. If it is an enzyme, we assign EC number, and if there is any evidence from different other tools supporting prediction, we also give the supporting evidence based on those matches, and apart from that, a unique identifier to the protein. So to start off with protein function, one moves on with pairwise alignments and does multiple sequence alignments. Does clusters and searches protein families within it, and also searches short regions of amino acids like the protein motifs, which carries out some functions. The other important terms which are related to assigning protein functions are the homologs, orthologs, paralogs, analogs. So homologs are two sequences which have evolved from the same common ancestor, but they don't share same function. So here's a picture of this definitions. I won't give the read everything. So in this picture, it's quite very easily and it's been easily drawn. So for example, orthologs are like they are having speciation here, and the gene is being transferred to another organism, and they have similar function. The paralogs, where the duplication event occur, two genes having similar function, and these are the xenologs, where same gene function but in different organism, and this transfer happens by the horizontal transfer. And another one is this one is the the paralogs, where it's called a neo-functionalization, where this gene evolved into a new function, and for example, myoglobin and hemoglobin. So to give protein a function, people use sequence similarity, sequence homology, and does functional homology. So so what does it mean? Sequence similarity means two sequence similar. Homology means they share the common ancestry, and functional homology means the two proteins from two different organisms have same function. So remember, like the functional homology doesn't guarantee the sequences are similar or sequences are homologous. So existing methods of doing function prediction using homology-based methods are BLAST, FASTA, S-search, PSI BLAST, and the PSI BLAST is like a kind of iterative method where it searches the weak hits also, and it identifies the domains and the motifs of the proteins. Subsequence method is based on protein domains, and there are some tools out there. The feature-based methods are based on the protein properties, for example, van der Waals volume, polarity, charge, and all these things, and they used to generate the descriptors of the proteins, and then you can run a classification model or some various other models to predict the protein function. What are the drawbacks are like, so for example, if you have a similar sequence and if it contributed to an error in one sequence, so that gets propagated to the next level. And also some highly homologous, so in typical BLAST and FASTA, the highly homologous sequences are not accepted sometimes at very high significance thresholds. So these are kind of drawbacks. So there are some enhanced methods developed from the Kihara lab, which integrates the PSI BLAST and the gene ontology terms together. So what it do, if you have a protein sequence, it does a PSI BLAST against the UniProt database, and against the PSI BLAST it collects the geoterms from their own developed PFPDB. It collects the gene ontology terms and they combine the scores using the formula here, as you can see, and it gives scores to the gene ontology terms based on the biological process, molecular function, and the cellular terms, and then each geoterms gets scored and you can infer the function of that protein. Sometimes homology searches fail. For example, no orthologs or even paralogs can be identified by sequence similarity, or they are unknown. No functional information can be transferred based on simple sequence homology. By instead analyzing the various parts that make up the protein, so it happens like, for example, sequence similarity or either homologous doesn't gives you good results. So people actually move on into the domains or try to identify motifs. So what are the methods for that? And they look for the domains of the various proteins in the eukaryotic and some other different kinds of species using different probabilistic methods like hidden Markov models and maybe some classification models, and try to assign functions to that. So one of the tools they use is they developed is the SMART, where they usually can give a protein sequence and it will assign the function. So SMART is based on a signal transduction elements of the protein, and this won't give you the everything for you. This is an example output of the SMART. And the PFAM is another very nice database where they have large protein families, and it helps, and also it contains the domain information. People search a lot in the PFAM, and the prediction is based on the hidden Markov models and tools, and also on multiple sequence alignments. This is another resource for doing searching the domains of the proteins, this CDD from NCBI. Now the resource what is good for me is, like as I told you, SMART is basically focused on signal transduction. PFAM is actively developed and is kind of up to date and has most up to date information of domain classification. There is another one, Interpro, which is which uses domains and assign functions based on the geoterms, and CDD uses the NCBI BLAST to search the protein sequences and then assign functions to them. So apart from using domains, sequence similarity, there are other methods like protein post-translational modifications. So what happens, like proteins get modified after the translation into, like, it added a myristoyl site, it added acetyl site, or it has some signal, it added some secretory signal peptide to it. So based on these different signals or the post-translational modifications, you can assign functions to the proteins. So two proteins might be similar if they have similar post-translational modifications. So the tool they developed is the ProFunc, where we can assign function based on the post-translational modification. So first you give an input sequence, then it does a feature prediction of the sequence, then it integrates the results into a neural network, and then it predicts the functions for you. This is the ProFunc server. It's quite popular, and it's pretty good also. And these are the results of the ProFunc. For example, if it's an enzyme and non-enzyme, so it will predict you whether it is an enzyme or non-enzyme, and then it will show you the results, the probability score, and also the odd score, and whether it is transport or binding, or whether it is cellular, it's present in cellular envelope, or it is it binds pyrimidines or purines or not. Since it's non-enzyme, so it doesn't have any enzyme class here. The ProFunc datasets are actually is based on Swiss spot sequences, which were categorized using the Euclid system. Enzyme category is also based on the Swiss spot, and the gene ontology terms are from the interpro. So sequences are divided into training and test without significant sequence similarity, and the binary predictors are given for each category of function, and then they're done to a neural network based prediction. So apart from the sequence-based methods, the other structure-based methods are like protein 3D structure-based methods. It's mainly based on assigning function from 3D structure. As you know, the primary structure of the protein, it gets folded into a secondary structure, which just further folding into a tertiary structure, and the tertiary structures combine to form protein domains. So all this information you can find it in PDB, SCOP, and CATH databases. The structure-based methods, if you see, they are homologous if they fold similarly, so they might function similarly. So there are cases where they are non-homologous. They have similar folds, they have similar function, and also homologous proteins, but they have different folds but having same function. So it's sometimes it's very tough to assign function to protein if this is the case. So they need to do a lot of this biological experiments, the microarray based experiments, to find out the correct function of the protein. The 3D structure based methods is based on the folding, the evolutionary relationships, the shape and the electrostatic properties of the proteins, the protein ligand complexes, or what type of ligand does it bind, and some molecular composition of the surface and the crystal packaging. So all these things comprise the 3D structure information from which we derive the protein function. So it's one of the very popular databases, and it's kind of, they say it's #1 for deriving protein function from the 3D structure. Is suppose you develop a 3D structure, now you want to search some functions based on the 3D structure, so you can use this and like, you just paste your sequence, and then it will predict the protein functions for you. And they have the 3D models built in at the back, and they will convert your sequence into a 3D structure, and then use the old models to assign function to your protein. There is also a video of this, this link where you can just see the live demo of the iTaser. Also, EMBL also came up with ProFunc, where you can just upload the PDB file, and then you can assign function uh, to the proteins. The ProFunc server is based on sequence-based and structure-based, and these are like templates-based methods where the function is being predicted based on these properties. These are the references for this lecture, and let me know if you want any further information on protein function prediction. I can help you out. Thank you. Hope you enjoyed the lecture.